Hi, my name is Narendra Suhas Jagannathan. I'm from the Duke NUS Medical School, Singapore, where I work with Professor Lisa Tucker-Kellogg in the Cell Modeling Lab. And today I'm excited to share with you a new work, TRANSCOMPP, a Markov modeling-based tool that we've used to estimate rates of stochastic phenotype transitions in heterogeneous cell populations such as tumors. Before we move on to what TRANSCOMPP can do and how it works, it's important to define this concept of cellular plasticity, which is the phenomenon where a cell can spontaneously or stochastically change its phenotype and display a new phenotype. Now, plasticity can occur both physiologically, for example, in the case of embryonic development or wound healing, or pathologically, as in the case of tumors. Under the physiological conditions, plasticity is most often seen in three different forms. For example, differentiation, where a stem cell can go to a differentiated cell type. Dedifferentiation, which goes in the opposite direction and can be found in selected niches like the intestinal crypt, or transdifferentiation, for example, the case where a differentiated fibroblast gives rise to a new differentiated cell type, for example, the myoblast. In the pathological conditions like tumors, plasticity is most often found in two forms, which can be epithelial mesenchymal transitions, which is the start of metastasis, and mesenchymal epithelial transitions, which is where you form a secondary tumor. And so plasticity in cancers could be of different types, like the EMT or MET that we've just seen, or it could also be through differentiation, which is linked with this concept of cancer stem cells, which are a small pool of cells that can differentiate and give rise to the bulk of the tumor. These changes during plasticity can be a result of mutations, or it can also be a result of epigenetic changes or post-translational regulation. The most important thing to understand here, though, is that plasticity can give rise to a very heterogeneous and dynamic tumor population, which can respond to cancer therapy in different ways. And so any anti-cancer therapy that you devise needs to attack not only the current state of the tumor population, but also all other states that are accessible to it as a result of plasticity. Plasticity has recently been linked to re-equilibration, which is a concept that's been found applicable to many different tumors. Re-equilibration is a process where a heterogeneous tumor population has an intrinsic steady state population composition, which could be a particular percentage of different types of cell phenotypes. And irrespective of how you perturb this heterogeneous population, you could even start with a purified population of one particular phenotype, the population always migrates towards the steady state. As you can see on the right in this figure from the seminal 2011 paper by Gupta et al., they purified three different phenotypic states from a tumor population and grew these states individually, and they found that over time, all three returned to equilibrium proportions containing all three states. It is entirely possible that a global phenomenon such as re-equilibration can arise from single cell stochastic decisions of phenotype, which is plasticity. And if we were to assume that these single cell transitions happen in a Markovian fashion, where a cell's future cell state depends only on where it is currently and not on its history, then we could think of formulating a Markov matrix or a transition matrix that captures the dynamics of this population and eventually results in this steady state re-equilibration composition. If we could model it this way, then given time series population data that captures how much percentage of the population is comprised of what phenotype, can we compute the set of transition rates or this Markov matrix that best fits this time series data? This is the question that TRANSCOMPP tries to answer. However, before we can move on to such transition rate computation, it is also important to understand some confounding factors that can make this computation a little difficult. What we observe in our bulk population data is the net transition from one state to another. However, it is composed of bidirectional transitions, and it is difficult to price apart how much flux there is in each direction. For example, we don't know exactly how much differentiation is happening versus how much dedifferentiation is happening. All we see is that overall there is X amount of differentiation happening in our bulk data. Another factor that can confound our transition rate computation is the fact that these different phenotypic states that we're talking about don't always have to proliferate or grow at the same rates. It could be that the stem cell grows at a very slow rate, whereas a differentiated cell grows at a much faster rate. And so this can also affect the overall population dynamics. And so ideally, we're looking for a method that can take into account these confounding factors like bidirectional transitions and phenotype-specific growth rates, a method that can work with both purified and unpurified starting conditions so that you don't always have to purify or sort each phenotype separately at the start of your experiment, a method that can work with unequal measurement intervals so you don't always have to measure periodically, and a method that can take into account all your different replicates and all your time points to give just one transition matrix that best fits all of your data set. And so we've developed TRANSCOMPP, a tool that uses optimization to solve for this best fit transition matrix that minimizes the fitting error between predicted and observed population dynamics. The advantages of TRANSCOMPP are that it does not require purified populations to start with. It can simultaneously solve for phenotype-specific growth rates if you're not aware of what they are, or if you already know what the different proliferation rates are, you can include that as a constraint during your transition rate computation. And apart from giving you the best fit transition matrix, it also gives you a confidence interval or a degree of uncertainty for your computed transition rates. And how TRANSCOMPP can give you these confidence intervals is based on a method called resampling that I'm not going to go into in detail, but it involves subsampling your data to get multiple pseudo data sets based on your original data set and repeatedly solving for the best fit transition matrix for each of these pseudo data sets. And when you do this, you get a distribution for transition rates instead of one single best fit transition rate. And you can use these to perform statistical computations, for example, get confidence intervals on or compare across different experiments. And so let me tell you about a couple of test systems that we've applied TRANSCOMPP to and the results we've obtained as a result. Our first test system is this MCF10CA1A cell line, which is a model for triple negative breast cancer. And so in this cell line, we use the levels of a biomarker CD24 to define two phenotypic states. A CD24 negative state, which is more stem-like, and a CD24 positive state, which is non-stem-like or differentiated. And so what we've done is we've purified these two different phenotypic states on day 0, and we've tracked these purified populations over time to see if re-equilibration really occurs. And as you can see in the plot below, irrespective of whether you start from a purified CD24 negative population, which is the red plot, or a purified CD24 positive population, which is the blue plot, the overall percentage of CD24 negative cells in your population moves towards a steady state value that's somewhere around 20 to 25%. And so this is re-equilibration, and this is what we would like to solve for to obtain transition rates that can fit this data set. And so we applied TRANSCOMPP to just the first 4 data points, and we observed that the best fit transition matrix would have the transition rates shown in this Markov model on the right. Stem cells had a 90% chance of self-renewal or giving rise to themselves, whereas differentiated cells had a 97% probability of giving rise to themselves. You can already see that there's a very small but non-negligible amount of de-differentiation that TRANSCOMPP predicts in this model. During the course of this experiment, we also noticed that the physically observable phenotype of your cells, which you see under the microscope, would vary depending on whether or not particular supplements were present in your media. These supplements were hydrocortisone, cholera toxin, insulin, and EGF, which are very commonly used. And so this raised a question for us. Are these supplements in fact also altering the plasticity of your overall population and biasing them towards particular phenotypes? And is that why you see these physical differences in cell phenotype? And so what we did was we repeated the time series experiment from earlier, but this time we added each supplement individually and then used TRANSCOMPP to compute the best fit transition rates. And we saw that yes, in fact, the presence of these supplements were altering these computed transition rates. We saw that hydrocortisone had the strongest effect, biasing the population towards a more stem cell-like population. Insulin and EGF also biased towards a more stem-like population, but had a weaker effect compared to hydrocortisone, whereas cholera toxin had the opposite effect and biased the overall population more towards the differentiated phenotype. And it was not just with the best fit transition rates that we observed these differences using different media supplements. We used resampling to get transition rate distributions for each of these experiments. And then when we compare the transition rate distributions that we obtained as a result, we saw that yes, there were statistically significant differences between these different media supplements as well. Hydrocortisone was definitely pushing the population more towards a stem-like state, and cholera toxin was definitely pushing it in the opposite direction. And both of these transition rates were very statistically significantly different from each other. And that was the first system that we applied TRANSCOMPP to, and we show that using TRANSCOMPP you can detect subtle changes in plasticity that could be a result of having different media supplements involved. And so now we move on to our next dataset, which is a published dataset of primary cells from patients with head and neck cancer. And again, there are two phenotypic states, a stem-like and a non-stem-like state. And this is a very long time series experiment where the authors tracked these two states over 100 days and had measurements for the population compositions every 3 passages. The authors again observed that there was a significant amount of re-equilibration happening irrespective of whether you start from a stem-like state or a non-stem-like state. And what we did was, given that this was a very long time series, we just took the first few data points and computed the transition rates using TRANSCOMPP and then tried to predict the future population based on the transition rates computed from these short-term data sets. And we saw that even using a very short data set, which would be modeled from just 3 points, which would correspond to about 9 days, you could reliably predict how the population would behave over a long term of more than 100 days. And so you could definitely predict long-term evolution from short-term data using transition rates that you compute using TRANSCOMPP. And so in summary, we've developed a new tool called TRANSCOMPP that can compute best fit Markov transition rates for stochastic phenotype transitions. And we've applied that to two systems, and we show two things. One is that you can detect subtle changes in plasticity using the transition rates computed by TRANSCOMPP. And you could also use short-term data to predict how the tumor would evolve over a longer time period. And so in this talk, I believe that I've given you a very concise explanation for what TRANSCOMPP can do and how it works, but a lot more details, including benchmarking, can be found in the associated paper that we published last year, and the paper can be accessed using the QR code on the screen. I'd like to thank all my experimental collaborators for both the MCF dataset and the HNCC patient-derived dataset, and all my lab members, the Tucker Kellogg lab, for their help with this project. Thank you very much.